recording here. All right. And
all set?
Yeah, all set.
Okay, cool. Well, thanks for hosting this, Mark, and for the introduction. Here, let me just share my screen real quick. Okay, can everyone see this? We all good? Okay, cool. So yeah, as Mark was saying, my name is Ben Cordier, and I'm a PhD candidate at OHSU in Portland, Oregon. And today I wanted to, I want to share a bit about some of our work we're doing towards quantum approaches for the computational biosciences. And I actually won't be focusing too much on quantum chemistry today. I'm actually going to take a little bit of a different tack, but I will kind of briefly describe some of those things. So before we begin, basic roadmap. I'll start by talking about the goal of our research and what has us interested in quantum computing, and a bit about why we're interested in quantum computing. Then I'll also be talking about some of the challenges in biology and give kind of a basic overview. And then I'll hone in on one specific problem, which is inferring mutational signatures. And I'll discuss a bit about the practical value of mutational signatures and what the use cases are. And then I'll discuss one classical approach to inferring them. And then rather than going into the details of a quantum approach, because this is a problem we kind of looked at but didn't end up really going with, I'll talk a bit more about what the potential is for problems like this one and looking forward. So what's the goal? The goal is to understand both theoretically and practically which computational problems in biology and medicine may be more tractable with quantum approaches. And so by theoretically, we're talking about kind of the formalisms in quantum information and quantum computing and the theory there. And we're also talking about the computational complexity and what that tells us about the types of problems that can be made tractable or more tractable by a quantum computer. By practically, we're really thinking about the translation. How can we translate this kind of from the lab to kind of the other lab, the biological lab? But really, you know, how can we translate this into practical use outside of the quantum computing domain? And by quantum approaches, we're thinking about mostly hybrid quantum approaches. And so these are approaches that use quantum computing to do some of the heavy lifting and use classical computing to do some of the simpler stuff. So it might be that the classical computer does some sort of machine learning or optimization routine, and the quantum computing is being used to create some sort of a very large state space that we can, you know, kind of compute on to find a solution. And by computational problems in biology and medicine, this computing is really central to biological research and medical research these days. So this is, you know, we kind of like work across this field, but we're thinking about things like problems in genomics, in proteomics, and in, you know, structure-based drug design and things of that nature. And along the way, we'd like to develop some proof of principles, but that's actually not something I'm going to be talking about today. So why quantum? There's some common arguments that we typically hear when we talk about quantum computing. One really kind of hinges on Moore's law, which I'm sure most people are acquainted with. Another one is a little more recent, which is that as the amount of data that we are generating as a society grows, in biology, we're very comfortably in the exascale now. How do we model and analyze that data more efficiently? Because classical computers are struggling. And then there's the other notion, which is maybe the original one, and this comes from the idea of the intractability of simulating physical systems and how quantum simulation may be able to do it polynomially relative to exponentially, which is kind of what we see in the classical regime. And then there's this more powerful model of computing argument. And then there's this other idea where we have, this set of tools and ideas from quantum information, computational complexity, and how can we use these to kind of start looking at hard problems in a different way. So I'll kind of talk about these three in a little bit more detail. So the first one is this data and scalability argument. And the idea is that scaling to very large data is increasingly challenging, and improving algorithms is one way to kind of partly solve this problem, but we also need to fundamentally rethink how we build hardware. And so this is something we're already seeing, especially in the space of, for example, deep learning, where we have moved from, CPUs, which was pre-deep learning, to kind of GPUs, which were one of the central hardware technologies that facilitated the revolution. And now we're moving on to TPUs and more robust classical architectures that can be more efficient. The other motivation is the physical intractability, and this was best encapsulated or described by Richard Feynman in the 1980s, but was also something that was discussed by the likes of Paul Benioff and Yuri Manin around the similar time period. But it's kind of summarized in this famous quote by Feynman where he said, "Nature isn't classical, damn it. And if you want to make a simulation of nature, you'd better make it quantum mechanical." And then there's this other set of ideas which we can kind of think as a generalization of Feynman's idea, which is that, well, it's not just simulating nature that quantum computers do more efficiently. In fact, there's this kind of whole set of problems that quantum computers can generate solutions for more efficiently. And in that respect, quantum computing represents a more powerful model of computing. And so unpacking this a little bit more, we have problems like prime factorization, which is conjectured to be hard in the classical regime, but we know because of Shor's algorithm is easy in the quantum regime. And I'm using kind of the term easy, meaning it grows polynomially with the size of the input to the problem, whereas hard, it grows exponentially. And this is kind of a coarse-grained measure of easiness and hardness in terms of computational complexity that is pretty common in that field. There's this other area, which is the quantum simulation, which is more efficient. And then in the top right, there's combinatorial optimization. And these are problems which we know are hard to solve to kind of the optimum solution, but that we can do pretty well with heuristics right now, but still we have trouble scaling them to larger problem instances sometimes. So that's kind of some of the motivations behind quantum computing. It's a more efficient model of computing. It can do simulation, physical simulation more efficiently. And it also might help us with scalability. And this is scalability is kind of a very forward-looking thing. This is not a near-term thing. And so what are some of the challenges in biology? And so one of the things I can't stress enough is biology is a very, very complex field. Behind the text on this slide, you see this pathway map, and this was generated by the people at Roche. I think the original version was generated in the 1960s. And this is a very, very small slice of the metabolic pathways in the human body. Biology is incredibly complex, and we often don't know what we don't know. And so having really powerful computational resources to kind of help us pull apart some of these systems is a really useful thing. So what are some of the areas that could, quantum computing could bring value to in biology? Well, we have simulating molecules and simulating molecular properties. And that's kind of something that's very useful for rational drug design, and specifically structure-based modeling. But there's other areas which are, mutational signature inference, which is related to precision medicine, haplotype inference, and multiple sequence alignment, which we often see in population genetics. We also see things like constructing phylogenetic trees. There's actually a quantum algorithm to perform that based off of Grover's algorithm. And these are all kind of relevant to other things too, like evolutionary biology and systems biology. But if we were going to say, well, what is the killer app for the biological domain? Much like, in chemistry and material science, it's really going to be the quantum simulation. This looks to be the killer app for the biological field. And so if we were going to map these biological problems into this kind of complexity theoretic framework, what we see is that these mutational inference and haplotype inference and multiple sequence alignment, these are the NP-hard combinatorial optimization problems. The simulating molecules is exponentially difficult, but can be done efficiently with a quantum computer. And machine learning is where we often talk about the scalability challenge. This is something that is classically comfortably in BPP and that we can simulate efficiently, but once we get to very, very large data, this becomes challenging. And so I'm going to focus in on this, this problem of mutational signature inference. But before I get into that, I'm going to tell you a little bit about what a mutational signature is. This is a pretty, complex idea, and I'm just going to try and give you guys a taste of what it is. But then I'll talk about a little bit about the applications. So the basic idea is, let's say you have a patient, and that patient has a leukemia, it's a kind of blood cancer. One way you could sample that cancer, because it's leukemia, is you could literally just do a blood draw. And let's say they have two different populations of malignant cells. We call these populations clonal populations. So let's say they have two different malignant populations, and then of course the normal cell population. We should get a roughly representative proportions in that sample when we draw it. And so then once we have that sample, we will look to see what are the mutations that are characteristic in each population. And so we see the normal has this kind of like these pink and blue bars in it. And those would be kind of considered the germline mutations. These are the mutations that you're born with. And they, if you live to a certain age, they're pretty much considered to be unharmful. This is the idea of we have kind of population level variation, which is very normal and it's what makes us each a little bit unique genomically. But then we might have these different malignant clones with these different genetic signatures. And I should say that when we talk about these mutations that characterize these malignant clones, what we're really talking about is chromosomal rearrangements, point mutations, copy number variations. And so this kind of gives you an idea that there's these different kinds of mutations and they operate at different structural levels. And to give an example, a chromosomal rearrangement, you could imagine an entire arm of the X chromosome, for example, breaking off and then binding to chromosome 7. These are large architectural changes in the genomic landscape of that cell. A point mutation is something much simpler. It's just a flip of the base, say from a T to a C on the DNA molecule at a specific region. And then a copy number variation might be that, say, a specific gene gets, or an exon of a gene, which is a protein coding region, that gets replicated multiple times. So you find that it gets overexpressed. So these are all different kinds of mutations, but we'll really focus in on the point mutations from here on out. So if we have these mutations and these different combinations characterizing a sample, we can then look to many similar patients, say all other patients with a specific kind of leukemia, and see what are the proportions of mutations that are shared across these patients, And among those shared mutations, is there a subset that we can use to kind of characterize what this cancer is and what is driving it? So that's the idea of the mutational signature. It's kind of a combination of mutations that we can use to characterize perhaps a subtype of cancer, or it might also be, for example, that we can use mutational combinations to associate them with a clinical outcome. For example, this person is likely to have resistant or leukemia that's resistant to treatment. So what are the applications? So one application, as I mentioned earlier, is precision medicine. And so to give you an idea of what the current status looks like, for most cancers, when we see them in clinic, we see that we have kind of the standard of care, a set of combination chemotherapies, maybe radiation that's used. And so we might start off, you're a person with a normal cell, and then once a cancer cell appears, this is kind of considered the genesis of the cancer. You have this initial driver mutation right here. And so following that initial mutation, this introduces genomic stability, and we have this, what's called a clonal expansion. These are new mutations that get picked up. And so you see this fragmenting of this kind of initial cell into multiple clonal populations. And so at this point, we might detect the cancer and target it with a first treatment, but that treatment may not be fully successful. And so let's say these two cancer cells or clonal populations survive, they'll go on to kind of continue their clonal expansion, maybe pick up some additional mutations. And so, this person might relapse then, and we would give them a second treatment. And again, it might not be fully successful. And ultimately, if we don't get successful with these treatments, what ends up happening is that we drive the patient's cancer towards a resistant disease. And this is kind of classical population kind of evolution, right? That we're putting a bottleneck on a set of mutations and the strong survive. So we've essentially strengthened this disease by treating it. And this is not what we want. This is very bad. So if we know what a mutational profile for our cancer is and we can match a patient to that profile, ideally we might be able to develop combination precision therapies that target that specific mutational profile. And so in doing that, we might be able to make sure that we actually get all the clonal populations and kill them all at once. And so that's one of the ideas. If you have these mutational profiles, you can kind of prioritize which precision therapies and small molecules should be developed to treat cancers. And once you have this kind of large library of therapies, you can combine them in a bespoke way to this patient to target the mutational profiles they have. Another use case is for early detection. And so, if you imagine you kind of have this like life cycle of the cancer, maybe not life cycle, but this kind of growth trajectory. And so you see the initial mutation right here, and then that kind of leads to this growth of a cancer here. And so ideally we would want to detect the mutation very early on, maybe here, maybe a couple of steps away, but that's what we'd like to do. And a patient, if we know their mutational signature, or if we have a library rather of mutational signatures, when we sequence a patient, we might be able to actually match some very faint signatures that are occurring in their body towards a known mutational signature. And in this respect, we're kind of looking at these mutational signatures as biomarkers for discovering whether a cancer is in the early stages for the patient. So that's early detection. So how do we find mutational signatures? How do we actually go through and discover them? And so I'm going to go through and describe one of the approaches that came out recently. And this is a combinatorial optimization approach. And so we start off with matched tumor and normal samples. And so the reason we do this is because the tumor samples they're going to be characterized by mutations that are somatic. And these are ones that you kind of accrue over your lifetime. And then the normal samples, there's going to be these inherent variations that are different from the reference genome, but they aren't actually the root cause of the cancer. And so what we want to do is combine these two sets of information and use that to call mutations. Now, I have this little blue box that says call mutations. I assure you this is a very non-trivial process, and it's a large analytics pipeline that's used. But the outcome is that we have these calls. We have these regions or areas of the genome where we say there's a mutation here, there's a mutation here. And so let's say we were to call mutations for many, many samples. And this is what the original authors of this work, there's a paper by Dash et al. That you can find on PubMed that goes through this process. They went through and they paired up these samples. So they had the tumors and normals for each patient. And they went through and called the mutations. And then what they did was they fixed the biological level of the hierarchy at the gene level. So you can go further down, kind of from genes to exons to codons to like the single bases of DNA. But genes are kind of the highest protein coding level. And so they fixed the gene level and they simply asked, is there a mutation on this gene or not? And they encoded it kind of as this binary, right? And then they went and used they generated 2 gene combinations, and they essentially tried to they used an algorithm to assign these. And so this algorithm is actually, it ends up being analogous to set cover, which is an NP-hard optimization problem. And it's specifically a weighted set cover. And they did this, and in doing this, what you find is that you can see that, for example, the 2 gene combinations that describe a cancer are, in this case, G1, G2, G1, G3, G2, and G3. And even though we see the 2 gene combinations also occurring in the cancer cells, because they occur in the normals, we can roughly infer that they are not the drivers of the cancer. And so this is a simplified example, but this is the general idea. And so in doing this, the authors are actually able to get pretty good results. They had a training set of about 5,000 tumors and normals samples and 1,500 tumor and normal samples for a test set. And they were able to get about 91% sensitivity on these 2 gene combinations in resolving which samples were cancerous and which ones were normal. And so before I go there, I should say that there's kind of 2 levels where we can increase the complexity of this problem. So in cancer, we generally expect that the number of driver mutations in a cancer is somewhere in the region between 1 to 10 mutations. And so here they essentially decided to go with 2 gene combinations, but we know that it can be up to a 10 combination that is driving a cancer. So that is, as the K goes larger, you get this combinatorial blow up, this, very large exponential term in the algorithm. And that's what makes this hard. Another component is that they started with the gene level, and the gene level is interesting, but we actually know that a mutation in one gene in a certain area can be beneficial or confirm greater survivability if you have a certain cancer, whereas a same mutation or a different mutation on that same gene can actually harm your survival. So going at the gene level is not actually the level that we would generally consider useful from the biological standpoint, though it is a very good starting point for this research. What we would like to do is go down to what we'd call the codon level. And these are the triplets of bases in the DNA that encode for an amino acid in a protein. So we have this K term, the size of the combinations that we allow, and then the biological structure where we have genes, codons, and then single nucleotides at the top. And right now, we see that classically, this is what's tractable with a CPU. We can get roughly to the gene level, and we can do it with a K of 2. The same authors had a follow-on paper where they mapped this over to GPUs, and they were able to get into the regime of having a K of 3, but they were still using gene level. And so the question here, if you want to map this towards quantum information in quantum computing is, well, so where does a quantum computer get us on this problem? We know this is an NP-hard optimization problem, but is there a gain in tractability that we can get with a quantum computer? Like, would it be somewhere here? Could we get to 4? Could we get much further? Could we get up to, say, a 10 combination at the codon level? That would be our ideal. And so this is kind of one of the questions we have is where does a quantum computer actually push us towards the spectrum of tractability for a specific biological problem? And so looking ahead, well, so I should say that in doing this, we kind of developed this first pass of this framework. And so we had this idea that we had the motivations for why we would be interested in applying quantum computation to a specific problem. And then we'd go through and look at the complexities of the classical algorithms that are available, the quantum algorithms, and what we could expect the classes they were in. And then we considered how would we make a minimum meaningful example of that problem to demonstrate a proof of principle, and how would we map this to a hardware device, specifically a quantum computer. And well, I'll leave it there. So if we go back to the complexity, what we find is that we can expect low order speedups for these NP-hard and NP-complete problems like the mutational signature problem. And so that tells us that we're probably not going to be able to get to that space where we can have a 10 gene or a 10 mutation combination at the codon level. That doesn't seem to be likely, but maybe it could be possible. I'll say a bit more about that in a moment. And if we look at the simulation, what we see is that we have the high order speedups, and these are problems that are hard. And so that's also useful. So going back to where we are now, kind of, looking ahead, we're currently in what John Preskill called the NISQ era. And so this idea that we have noisy intermediate scale quantum computers. And these are small quantum computers that are, well, one recently demonstrated the first example of quantum supremacy. But they're noisy and they're especially quite constrained in the size of a problem you can map into them or in their representation. But there's a lot of different companies working towards this. IBM recently released their 53 qubit device. Intel is currently working on a 49 qubit spin qubit chip in silicon. Rigetti, which is one of the startups, they have many architectures available, and they're productionized through a public API, and they claim to have a 128 qubit system in the works. Google demonstrated that quantum supremacy result recently, and they are currently prototyping a 72 qubit device, reportedly. D-Wave is kind of the long-standing entrant into this field, and they actually have a quite mature quantum annealing device now, and they're about to release, if they haven't already, a 5,000 qubit quantum annealer with over 3,700 or 37,000 rather couplers, which is kind of the connections between those qubits. And as Mark said a couple of weeks ago, Honeywell soon will also be deploying a state-of-the-art ion trap device, and they claim to have a pathway towards scaling this which is something that's very exciting. So these quantum computers are small, and one of the, with the mutational signature inference problem, as I mentioned, it's a combinatorial optimization problem. This is a hard problem, and one that we don't expect we can get a lot of benefit from a quantum computer unless we have a very large quantum computer. But it may be possible to do some proof of principles in the near term, and one of the ways we would consider doing this is with a variational approach. And so the basic idea here is that you have a problem and you kind of leverage your domain knowledge of that problem to devise a cost function, and you include that into a Hamiltonian, which kind of describes the dynamics of the quantum system. And then you use that, you have this set of parameters theta, and these are essentially used to parameterize a quantum circuit. And so you parameterize a quantum circuit, and then you run it, and then you measure the output, you measure the expectation of those parameters, and then you calculate the energy and adjust those parameters again, and you kind of cycle through. And so you can kind of think of this as being kind of an optimization or machine learning problem on the outer loop, and then a quantum circuit on the inner loop. And so this is kind of what we mean about letting the quantum computer do the heavy lifting and letting a classical computer do the stuff that it knows how to do really well. And so this is an approach that we could potentially map to a problem like the mutational signature inference problem. And so that said, I wanted to kind of describe some additional considerations around variational approaches. And so as I kind of mentioned, they allow us to use NISQ hardware to tackle problems that would be impossible in the near term. And so we see this with, we're already seeing this with quantum simulation. There's a lot of examples of researchers using variational approaches to simulate molecules like lithium hydride and other different chemistries. We also have seen examples for quantum machine learning and also optimization. But it seems that a lot of these approaches are still fundamentally limited, and a lot of these limitations can be put down to the constraints of NISQ devices. The other thing is that if we look on how to do a variational approach, it's still a relatively non-trivial thing to do. So there are a lot of libraries that have been developed and are under development, especially Python libraries. But even if you kind of want to roll your own variational approach, you do need to have a deep understanding of the problem you're trying to solve with one. And so it's going to be very critical that if people want to develop these variational approaches, we think that interdisciplinary collaborations will go a long way to kind of bringing this stuff into the domain sooner. And then, well, I won't talk about there's some lessons from machine learning research, but that kind of goes into the weeds, and I won't talk about that. So this is actually a little bit of an older slide, but it's this idea that, how do we get from NISQ to fault-tolerant quantum computing, and what does that look like in terms of applications? And so we saw in 2018 that 53 qubits was sufficient to demonstrate the first quantum supremacy. I'm sure there'll be other examples of quantum supremacy to come. I don't think this was a single moment, rather it's probably, it's going to be a process. And then, with more than 53 qubits, can we actually start to develop early proof of principles and maybe even demonstrate some quantum advantages on specific domain problems? And then as we scale these up, can we get near-term applications? And so we see that, we need kind of lower error rates and more qubits. There's this, one of the reasons I say this slide is old is because on the right, we see this region of kind of 1 million qubits and a 1 in 1,000 error rate. We can start to see useful error-corrected quantum computers. And this is kind of the region of fault-tolerant quantum computing. But it may be that this is actually less optimistic. There's been some recent research where there's been recent research that has shown, well, there's the Honeywell example. So they claim to have a scalable ion trap device. The other thing is that we think error correction now may be possible sooner, and this is to do with the idea that there was recent research where they could map, Benjamin Brown showed that you could map a 3-dimensional service code to a 2-dimensional chip. And in this way, you could use Clifford gates plus a non-Clifford gate, which gives you the combination of those two, gives you universal quantum computing. And non-Clifford gates were thought to require a process called a purification process called magic state distillation. This is something that's used to extract the errors out of a quantum system. But by showing that this mapping could occur, it might alleviate the need for that. And so that would mean that we could see error correction in fault-tolerant quantum computers much sooner. So all that said, the upshot is that, at least for biology, the simulation of molecules we think is still going to be the killer app, right? But we also see lots of potential for applying quantum computing across other areas in biology. And so mutational inference, signature inference is one example of this, but there are also many other problems. And they could, especially, there are many problems that could make use of the variational approach, which I described earlier. And in that spirit, heuristics that incorporate domain knowledge will be central for success. That's where we see a lot of the potential benefit coming. One of the challenges is we can't throw these heuristics cleanly into that computational complexity hierarchy. So it really is kind of this, what we really need to do is just try. We need to try and see if these heuristics, these variational approaches will work and give us good solutions. And so part of that, means that we need to come up with very good validation procedures once we get solutions. How can we verify that they are actually correct? And to really bring quantum computing into the biological domain, it's our view that interdisciplinary collaborations will be required. And so this is something we're actually working on right now. And then, like it's early days, but like I said, I think there's a lot of reason for optimism. And so with that, I'd like to say thank you. I don't know if I went over under time, but I'd also like to say that our research is a collaboration between OHSU's Department of Medical Informatics and Clinical Epidemiology, which is where I am, also the Knight Cancer Institute, Portland State University, and Intel. Yeah. And with that, I'm happy to answer any questions too.
So thank you so much, Benjamin. So yeah, I've sent a note to everyone. If anyone has any questions, please include them in the chat box there.
Yeah, absolutely.
And for those who asked, yes, we're recording and it should be available by tomorrow. So there's a question, are you working with Intel's quantum computers?
So no, we are not yet. And we're in the very early stages of the collaboration with Intel's quantum team. And we are currently just kind of establishing what our direction is going to be. But it's probably actually going to be more in algorithms research rather than working with their hardware.
Good. Are there any other questions that people have? Could you name some Python libraries that you referenced?
Oh yeah, sure. So IBM has one, Qiskit, and that's a good one. And they have a very robust kind of ecosystem that you can check out online. And they allow for all sorts of different things. I mean, you can do kind of modeling of noise and on the chips. You can also do some quantum chemistry and some algorithms research or work with it. And you can also do kind of low level like compilation and optimization stuff. There's also, Google has a library called Cirq. And Rigetti also has, they have Forest, an API to use. quantum machine learning, I think there's a Penny Lane, right? Is that correct? Yeah.
Yeah,
And so,
that's by Xanadu, a group in Toronto, has Penny Lane.
yeah, it's Xanadu, right?
Xanadu, yeah. Yeah, they name all their products after Beatles songs.
Yeah, great taste. Yeah. So yeah, there's a lot of different libraries.
Great. Have you figured out what the Hamiltonian would potentially look like for a problem like this?
No, we have not looked into that yet. And in fact, so the mutational signature inference is something that we were looking at previously, and we actually were thinking we might not pursue that specific one, even though it's quite a nice problem.
There's a question. You mentioned that there's a way to do phylogenetics with a modified Grover's algorithm. Are there algorithms that you've begun working on for mutational signatures outside of the general variational approach?
Sorry, was it the phylogenetics?
Oh, maybe it's a typo. Maybe they meant P.
So,
yeah, typo, yeah.
So, sorry, could you say the question again, Mark?
You mentioned that there's a way to do phylogenetics with a modified Grover's algorithm. Are there algorithms that you've begun working on for mutational signatures outside the general variational approach?
Yeah, so the phylogenetics, this is actually kind of an old publication, and it was specifically an approach to, there's an approach to kind of building out the phylogenetic trees. That is an algorithm which probably will require a much larger quantum computer to perform, but it is interesting. We're currently exploring problems. We're looking at quantum simulation because we really do think that's going to be the area that is going to move first and probably going to have some of the most exciting developments in terms of the biology. Yeah.
Super. So if there's no other questions, I guess that's it for this one. In 2 weeks, we will have Jennifer Pakin from Galois talking about formal methods. So that should be great. Super. Thank you, everyone, and I'll see you again in 2 weeks. And thank you especially, Benjamin.
My pleasure. Thanks for coming, everyone.